There's many processes that are marketed to try to help measure concussions and help with recovery and get people back on the pitch, but the vast majority are not validated. So as scientists, it's very hard to rely on them. And as a clinician, it's even further harder. So part of that objectivity that I want to bring to you is that theme is being able to take from the lab to the pitch, to the clinic, objective measures to get someone back safely. We know that 90% or more of concussions recover in 7 to 10 weeks or days, but you know, there are catastrophic events. Those of you from Ontario know about the Rona's Law that developed after the poor girl in Ottawa died on the pitch in high school. There was a recent death in Premier Rugby in Australia, and so forth. So there are deaths that occur. You know about, you see in the paper about all the variety of athletes that are suffering from chronic traumatic encephalopathy. So this has real consequences. So I think there's real value, and it's a great pleasure to bring this panel of experts together today to be able to provide that. Each of them are going to introduce themselves, and I'd like to just bring up Dr. Patrick Neary, who is going to lead us off in this adventure.
Thank you, Steve. Can everyone hear me all right? And you are right about this light. I might stand back, and I'm hoping that this can you can still hear me. As Dr. Martin just mentioned, we're all very passionate about concussion and whether or not we can actually not only diagnose but maybe help in recovery and management of that. And that's really how this presentation came together. 2 years ago, we actually put together a conference also in St. John's, Newfoundland. And when we weren't on George Street, we were talking about concussion. And again, we felt that we needed to get the information out. What is actually happening in this area of concussion? Are there biomedical, are there biomarkers that we can actually use to try and get some indication of what is the stage excuse me, that an individual is actually at? And so that really led to what is actually happening and what are the prospective studies. And so our whole focus today is really to look at that. Not only the objectivity, as Dr. Martin just mentioned, but also what are the prospective studies, because the audience back in St. John's, that's what we're asking. It's okay to look at day 1 versus day 30, but what's happening in the middle? And so that's what we're going to try and focus on today. And my aspect is certainly just to look at the cerebrovascular aspects related to concussion and what are the mechanisms involved. And the major areas that I'm going to cover today look at cerebrovascular reactivity, and I'm going to focus in on that. We're going to talk about neurovascular coupling as well. And then we'll also talk a little bit about the cerebral autoregulation and move forward with that. Again, trying to maintain this focus of prospective studies. So we were fortunate a number of years ago that we had one of the first studies to come out to show that certainly, indeed, we do have changes that are going on in the brain from a cerebrovascular reactivity perspective. And what we actually used was a very simple test where we used carbon dioxide and simply using a breath hold challenge to create a hypercapnia. And we collected baseline data for 5 minutes, and then we had our subjects go through a 20-second breath hold followed by a 40-second normal breathing. And we did this 5 times. And then we also did exactly the same time and created a hypocapnic challenge where we did a hyperventilation to try and create this effect within the brain of a cerebrovascular or CO2 reactivity. And what we actually showed was over time, day 2, day 4, day 8, when we take a look, we found that there were significant differences from day 2 to day 4 to day 8 from the baseline. And these were transient effects, and they got better over time. And this happened both with a breath hold challenge, as I mentioned, the hypercapnic, but also with a hypocapnic challenge. When we looked at the changes in cerebral blood flow in relation to the CO2 challenge we were giving the body. And again, we found significant differences at different parts within the protocol that we were looking at. And again, simply just a breath hold or hyperventilation. So we concluded from this that certainly under resting conditions, we weren't finding differences in the cerebral blood flow. So normal and rest seemed to be very, very similar. So there wasn't a challenge to the brain. So the brain was able to handle that. And we also were able to illustrate that this was very similar to others that have published as well, looking at heart rate variability. Sorry about that. Go back. We found, though, that the CVR was impaired from rest to day 2, getting better on day 4, and certainly better again on day 8. And again, that really confirmed some of our original research to illustrate that there really is this impairment. And in our first study, we simply looked at rest versus day 4, and that was it. We didn't have the prospective data at that time. And then certainly, as I mentioned, there's a significant effect on day. And then we were really excited that Mike Ellis out of Winnipeg, as well as Alan Much, started looking at the brain using imaging, but they also used a really, really neat device called a Respiract. And what it actually does is it blocks your CO2. So regardless of what your ventilation rate is, it keeps your end-tidal CO2 exactly the same. So then Alan and Michael and his colleagues in Winnipeg were able to illustrate, using these MRI maps, there are problems associated with the brain when you're looking at a cerebrovascular reactivity. And so they concluded that certainly the CO2 is extremely, extremely powerful stimulus in order to change the blood flow and blood flow dynamics. They were also were able to illustrate that very, very small changes in PCO2 could create up to about a 15% change in your cerebral blood flow. And then again, just wanting to really lay down this idea that the CO2 is a very, very important mediator of what's going on in the brain. So if there's some way that we can use something as simple as CO2 in order to get an indication of where our athletes are at, and especially as Dr. Martin mentioned, you've got somebody that's been knocked out on the pitch, but 5 minutes later they seem to be walking around and seem to be coherent. So what are some of the other prospective studies? More recently, there's again been some more MRI studies where they've looked at very, very specific regions of the brain. And in particular, the mid-insular region, the sulcus region as well of the brain. And what they've been able to demonstrate here on day 1 versus day 7 or 1 week versus day 30, what are the actual changes that are going on from an MRI perspective? And in this particular study, and I apologize for the size of this, they first wanted to look at behavioral modifications and behavioral changes. And were these athletes anxious or were they depressed? And the research that they showed was in relation to the healthy athletes. On day 1, they were able to show that, yes, certainly, there are some changes in the anxiety levels or depression levels. And this held true and seemed to get better as well as time went along. And by day 30, they're basically back to normal, or in this particular group. But you can also notice from this slide that there's a great variability in this. So the severity of the concussion also makes a significant difference. So we'll talk about that in a moment as well. And then, of course, they looked at this regional blood flow based on the MRI information. And again, it showed a very, very similar pattern in that here's the healthy, all right, at day 1, day 7 or 1 week, and then at 30 days, we've got this U-shaped effect. And so there were significant differences in these different particular regions of the brain. So what does that really mean, though? When Dr. Bjornson gets up and talks as an MRI specialist and a clinician looking at pediatrics, is it necessary for your children or my children to have an MRI? And what information will that give him or is it better for us to look at the research and then try and provide that information for clinicians? So again, there was a relationship between, again, some of the symptoms here as well as the cerebral blood flow measurements that they were able to look at with the MRI. And then they actually divided their group into those that had a good outcome versus those that didn't have a good outcome, and looked at, again, the relative changes in their cerebral blood flow. And again, they were able to show that there are some significant differences depending on the day. So over time, you can see that this is certainly getting better, but the first couple of days after a concussion is an important period. And it's one that we as researchers are trying to adhere to. If we can get them within 1 to 2 days, that's the best for us to get some information. And then to see what happens prospectively over time. And so again, depending on the region at which we're looking at and what we're trying to get an idea from, we can find that there are differences in those who are recovering well and those that are not recovering as well. So from this, they really stress the fact that individualized data is important. We are all individuals. We all have a different physiology. We can all be hit with a different amount of, or the same amount of force, and end up with a different severity of concussion. So that was important. They realized that there are perfusion differences and deficits that are occurring, both longitudinal and cross-sectional. They were able to show that there was reductions after day 1, getting better by day 7, but certainly better by day 30 as well. And then again, they felt that the perfusion was a problem, which now comes back to, what is happening with that interplay between the arteries and the cells. And again, I know that Dr. Gaetz will talk a little bit more about some of the electrophysiology involved. And one of the limitations to this study, though, is that there was no baseline data. And we really believe that it's important to get the information before the season starts, if you can do that. It's not always possible, but if you can, that's the best for you to be able to then compare what's happening over a week or 10 days or 2 weeks of a concussion, and over the season as well. Another study that's come out more recently in this last year is they actually have done MRI using soccer players in high school. And what they actually did is they used an interesting system called an X patch. And here it is up here. And if I can get the pointer, it's a band that goes across the forehead. And there's also a little band or a device that's placed in behind the ear. And what this does is it records the acceleration when they're heading a ball. And so what they did was they actually then looked at the MRI results from a whole season of soccer and high school football or soccer. And what they were able to actually then illustrate is, let's use a particular measure of the MRI to give us some indication of the vascular reactivity. So we're using some very sophisticated equipment to look at the cerebrovascular reactivity and what's going on over a season of soccer. First, just to illustrate what happened with the acceleration data, or the force data, if you will, they then divided their groups up into those that did a lot of heading and those that didn't do as much heading. And they plotted that over the season as well. And you can see here, within about 5 weeks, within about half of the season, and then again at the end of the season, you can see that there's really a linear increase in the amount of force that's being applied to the head in female soccer. And this is high school. And whether or not it was this translational or whether it's angular, they illustrated the same sort of effects that are going on. So then what did they find with the actual cerebrovascular reactivity data? And does it match this? And actually what it does is it matches it very, very well. And they were able to show that there are changes from the baseline. And that's what this is here. They're looking at a functional change from the actual baseline and using this measure of a beta weight from the MRI to give an indication of the reactivity. And they were able to show that at 1 to 2 months, all right, 3 to 4 months, we still have changes in the vascular reactivity within the brain, and this seemed to coincide very well with the amount of force that was applied. And so they found a good relationship between those. So again, what they concluded was that during a competition, all right, there is decreases in the athlete's cerebral vascular reactivity, and we really have to take a look at this, especially between the beginning and the end of the season. Between the beginning and the end of the season, there was also a cumulative loading that was going on with these young female soccer players. This cumulative loading really suggested that we need adequate rest at the end of the season. But let's not fool ourselves. Today, these young athletes are participating in anywhere from 10 to 12 months. So if you start out with a concussion at the beginning of the season, and it's not completely resolved, and prior to this we were just talking about Sidney Crosby and whether or not his initial concussions were completely resolved. And it'll be interesting to see when he actually comes back to play this year as well. So then we've certainly been able to illustrate that, yes, there are the cerebral vascular reactivity problems. There are the brain physiology that we can actually monitor objectively, and something that Dr. Martin really wanted us to speak to. More recently, we've also conducted some research to look at neurovascular coupling and what's going on in the brain when we do that. And this is some unpublished data that we have from our lab, but it has been presented. And what we actually did was we mounted a variety of pieces of equipment, a transcranial Doppler. We use a Finipress for blood pressure, continuous blood pressure. We also have, you can see here, respiratory measures, gas measures, so that we can get our end-tidal CO2 values. And what we do is we put them through, again, a very simple protocol, what I've already described, looking at cerebral vascular reactivity with a breath hold. And then they went in and they actually did this neurovascular coupling exercise where they did a task switching. There was a computerized program. There was a red dot on one side, and it moved back and forth. And you had to use your finger in order to try and get the brain to operate to find out what side of the computer screen was going off. And so we presented this in San Francisco a few years back, and what it actually illustrated was that, again, when we take a look at the blood flow in relation to end-tidal CO2, we found there were no carbon dioxide changes over this period of time, but there were cerebral blood flow changes, which indicates that reactivity has changed. And the first one here looking at resting baseline, very, very healthy individuals. And you can see that as we move along from this 20 seconds of no breathing and then the recovery period, that we are finding differences in the reactivity over time. We can then take a look at day 4 to day, whatever that is, day 6 or day 9. We can move beyond and take a look at day 10 to day 15. We can then look to see when they start to come back, and these individuals on average went back to play somewhere around about day 16. And we can find that over time, this response is now coming back to normal. But whether it comes right back to normal is really, really difficult for us to look at. And actually, it's easy for us to look at, but in order to state whether or not it's statistically significant or whether biologically significant is really the tricky thing that we're trying to work on as well. And as I mentioned, there were no differences when we take a look at end-tidal CO2 values, which just really confirms that if there are changes in cerebral blood flow, no changes in the end-tidal, then obviously reactivity has to be changed. When we looked at the neurovascular coupling test, what we find is that a very, very similar result. By day 1 to day 3, we're finding big changes, huge changes in that. It's starting to come back to normal, day 4 to day 9, and then again getting back to normal when we return to play. And so we concluded that certainly over the first 3 days that we have a blunted response. Is there anything that we can do during that first 3 days? And that's the question that we're trying to ask as well, based on this type of information. I'm just going to move forward a little bit quicker here because I know I'm running out of time. Neurovascular coupling responses, again, in relation to that neurocognitive test. were able to illustrate that there was a lower CVR in these particular athletes with a sport-related concussion. And again, we know that this starts to resolve over time. And again, the emphasis here and the important aspect is to note that cerebral blood flow will change. It does change over time, but when it's blocked to CO2, we didn't find that the CO2 did change, indicating, again, cerebral vascular reactivity as well as the neurocoupling changes. And again, I need to emphasize here the subjectivity of this and being able to provide objective measures. We also collect subjective information, the SCAT scores, and that's the Sport Concussion Assessment Tool. For those of you that are doing any work related to concussion, you'll be well aware of this. And so we actually plotted that as well to see what the actual changes were in relation to the objective measures, and certainly the timeline between the two are not the same. Athletes, I'm sorry, will lie to you. No ifs, ands, or buts. All right, they want to get back to play. And their symptoms seem to come down a lot sooner than what the physiology is showing us. So you have to be very, very cautious. More recently, we've continued to investigate this reactivity, heart rate variability, blood pressure variability, and we've just had this accepted this month looking at, again, blood pressure and heart rate and what's happening during the very, very acute stages of this to illustrate, again, that the brain is very, very susceptible during that first 72 hours. We are lucky that we worked with a number of the Western Hockey League teams. We've got a huge cohort of anywhere from 89 to 140 subjects, depending on what particular subject pool we're looking at. Again, looking at the type of equipment that we're using, transcranial Doppler, the respiratory measures, blood pressure, ECG as well. And then we go into, in this particular project, we used a squat stand. Our previous one, we wanted to perturbate the brain using CO2. This time we wanted to perturbate the brain using a pressure drive. So we used blood pressure to drive the brain to see what actually was going to be happening. And again, what we were able to illustrate was that the heart rate itself wasn't statistically significant, but when we took a look at the standard deviation of the heart rate, we did find that there were significant differences, whether they were in the squat phase or whether they were in the stand phase. We then followed this up and looked at our blood pressure data, and again, we can take a look at the squat phase, the stand phase, and we were able to show the differences between the concussed and the mild traumatic brain injured as far as the pressure drive to the brain, and significant differences here. So there was a significant decrease in the late phase of this stand. And again, we can tell that something is going on. The brain is having to deal now with a pressure drive to the brain. And how does it do that? What are the auto-regulatory mechanisms? And how are they affected? And so from this, we were able to conclude that certainly there's this baroreflex that is going on. It seems like there's a pressure alleviation, and in other words, there's a protective effect. We found that change in the standard deviation, or a very, very small change in the standard deviation with those who have concussion in relation to those that are healthy. And then again, we felt that, again, looking at this all together, there is this autonomic nervous system dysregulation that's going on. This was then followed up with some other research that's recently come out of the literature, and they wanted to really determine whether or not is there an autonomic nervous system dysfunction that is actually happening. And they used a grip strength dynamometer in order to elicit the sympathetic system. And as you know, when you perform high-intensity activity, you get a sympathetic drive. So can we use a sympathetic drive to try and tease out whether or not there are autonomic dysregulations going on? And what their results showed, and I'll just go through this quickly to illustrate that at 48 hours, right here, 48 hours and 1 week, they were able to show that when you take a look at this particular waveform, the blood pressure waveform, And you'd look at the time in between these two particular waves, or peaks within the wave, and they call that their cis slope, rise over run, and they've suggested that this is a measure or a surrogate for stroke volume. They illustrated that there were significant differences between both the controls and the concussion at 48 hours as well as at 1 week. So even after 1 week, there's a suggestion that the autonomic nervous system is still not working at par. They then continued to not only look at this stroke volume index, but they also took a look at the geometric shape of this to try and get an indication of what is actually happening there as well. So not only the timing, but also the shape of these contours. And again, they were able to illustrate after 48 hours as well as after 1 week that there are significant differences that are happening. So they concluded that certainly there are, go back real quick here, if I can get back, certainly they did conclude that because of this, that there are autonomic nervous system problems. So we need to be able to deal with that, but how do we deal with that? And that's the area that we're coming to now. And we were able to confirm this using, again, a specific device in our lab called seismocardiography. It measures the seismic activity or the mechanical functioning of the heart. And we were able to show that there are timing differences both between a concussed and a control. And although this is not prospective data, what it does is it really confirms that there is this autonomic dysfunction in individuals with a concussion. And this is just the seismocardiogram, and what it actually shows you is that we can look at mitral valve open or end diastolic volume, aortic valve open, etc. So it's an interesting device that we can look at from a cardiac or a cardiac cycle perspective. And more recently, there are individuals that are now starting to look at near-infrared spectroscopy, looking at the oxygenation within the brain. We've been able to show blood pressure, blood flow, heart rate variability, but what's happening to that oxygen? And as you know, every cell in the brain, every cell in the body needs oxygen in order to function properly. Unfortunately, this study was not prospective. It just illustrated that there are differences in oxygen levels between the healthy individuals and the concussed individuals. So we followed this up, and a master's student of mine followed this up to look at what is going on with prefrontal cortex. And Scott actually presented a poster earlier today talking specifically about that, looking at prefrontal oxygenation levels with near-infrared spectroscopy in concussed individuals. And just to summarize very, very briefly, we continue to use our CVR CO2 stress test that we're very happy with. We used the near-infrared spectroscopy. And so what did he find? Well, we did find that when Scott plotted his data and took all of those 5 intervals and plotted them within a 1-minute period, was able to plot to look at the differences between rest and the breath holes. And there are some differences between the controls and the concussion here, although you can see that we are starting out at a different level. And this is the relative change, going from rest to peak value only after 1 to 3 days. We then replotted this data to look at what's happening over time. And certainly over time, we're finding that from the control to 7 to 14 days, we're finding that we do find some statistical differences. One of the most interesting findings I think that Scott found from this data, however, was the fact that when we looked at the standard deviation of the oxyhemoglobin the oxyhemoglobin value, we found that there are significant differences from the resting value through to day 1 to 3, 4 to 6, and comes back to normal after the 7th or 10th day. And if you take a look here, you can see that, again, this standard deviation in relation to the control value is significantly reduced. It's half. And so, again, something we're finding is that the standard deviation of the measurement is really, really important. In other words, they're operating at a reduced limitation. Their electrochemical variance is significantly reduced. So again, just reinforcing the fact that these changes are happening in the brain, and we really have to be cognizant of them, and certainly no pun intended. Finally, the last slide here is there's some, I'll just go back quickly here. There's some individuals out of UBC, and some of our colleagues actually have looked at this in another light. They've taken some MRI and they've looked at the white matter and white matter changes, whether or not there are lesions that are happening in the brain over a full season of ice hockey. And probably the most important finding here is to show that when they divided their individuals into concussion versus non-concussion, they found that after 2 months, they found, and again, I'll have to ask Bruce more about this because his brain is better than mine when it comes to MRI, but a 0.23% reduction after 2 months in their brain volume changes. And so statistically that was significant. Whether or not that has a biological significance, I have no idea. But what it was trying to illustrate here is that after 72 hours, there wasn't a change. After 2 weeks, there really wasn't a change. But again, after 2 months of ice hockey and having a concussion, they illustrated that there were these changes. And so they concluded that no brain volume changes really that were detectable after 3 days and 2 weeks, but some of these changes were actually detected after 2 months. And they did find that there were no detectable microbleeds from the concussion during a season of ice hockey. So that was good to know. And more recently, some more prospective data that's just coming out from Mike Ellis' lab, and Alan Much have illustrated that we really have to look at these studies over time, longitudinal, as well as compare it with cross-sectional data. So the final summary that I have here for you is looking at both cerebrovascular and cardiovascular changes, blood pressure, heart rate variability. The standard deviations of these measurements are also extremely important from the data we've collected in our lab. We've illustrated that there are alterations related to the autonomic nervous system. It's a transient effect. It comes back to normal. When it comes back to normal, well, we don't always know. And then again, severity is also problematic and something that we also have to look at in more detail as well. I'd like to thank my collaborators and some of the individuals who have helped to put this together, as well as our funding agencies. And I think because of time, we're going to allow Dr. Gaetz to get started, and then we're going to have a panel discussion at the end.
Well, thank you for letting me be the creamy white center of your concussion Oreo today. I'm a little concerned that Patrick had that statistic about a 0.23% loss in brain matter with 2 months of hockey. I was doing the math over there, and after 40 years, I think my brain's probably going to start to rattle by the end of this talk today, so hopefully that's not actually what's happening. But so what my talk is going to be about today is, it started out 6 months ago when we put this symposium together. I was going to focus on electrophysiology. That's my background. But more recently, I've been looking at and studying and thinking about other things related to concussion, including CTE. So what I'll be doing today is looking at prospective science in that area, in CTE research, but also electrophysiology, which is my area. So, and I'm sure you're aware that this is very topical. It's something that you hear about in the news regularly. This image actually is from a PBS broadcast Frontline about CTE and about some of the brain changes and stages that have now been proposed to describe CTE pathophysiologically, but also from a symptoms perspective. So this image is from CTE stage 2. You start to see some pathophysiologic change in the brain, and then there's some correlate of some symptoms, rage, impulsivity, and depression, that have been linked by some to that stage. So this is what I hope to accomplish today. We're going to look at chronic traumatic encephalopathy. Some of you know what it is. Some of you have heard about it, but might not be exactly sure what it's about. We're going to look at the science behind CTE. So given that we hear about it all the time, daily in the media, in science, one would hope that the science behind this area is robust. We'll look at prospective studies in CTE science. Then we'll shift our focus and look with an eye to my area, electrophysiology, and break it down into EEG, evoked potentials, and event-related potentials, 3 different ways to look at the brain electrically, and then focus on which electrophysiologic studies are prospective in nature. Okay, so with prospective research, it's sort of the overall governing theme of these talks is we want to do prospective science because one day we hope to be able to move from working with groups to working with individuals, and we're trying to come up with sensitive measures to assess and manage injury prospectively. So with prospective work, we're looking to the future, we're looking for a change in an individual that's related to something specific, and in this case, concussion. So prospective studies are going to monitor the outcome within a study group as it relates to a suspected risk or some preventative factors. So this is what we hope to do with the area of science that I'm in and Patrick's in and Bruce is in. We hope to come up with some sensitive measures that we can see changes within individuals and one day come up with some clinical criteria based on those measures. So, CTE in the news, these are the things that we've been hearing and seeing and thinking about over the past few years. You've seen it with lots of high-profile athletes, actually, who've passed on. Here's one example from the NHL, where an NHL enforcer, Derek Bugard, died, and his brain showed evidence of this chronic traumatic encephalopathy. There's many examples from the NFL, where NFL players following retirement have passed on either by suicide or cardiovascular disease or some other disease state. And if you look at their brains are donated to brain banks for study, and if you look at their brains sometimes, as in this case with Junior Seau, there was evidence of CTE. You've all heard that there was a large lawsuit initiated by some players in the NFL, and the lawsuit was settled, and it was decided by some that it was too low, and they were thinking about going back to court and reopening the case, but recently they've decided that a $1 billion settlement is going to have to do. So that was this year, so this is ongoing and recent information in the media. This is one from this year as well. This was a headline in the Washington Post, the terrifying link between concussions and suicide. It's very topical in the CTE literature today, topical and controversial. Essentially, what certain folks in the media and some scientists are saying is that with every concussion you have, this increases the rate at which, not the rate at which, but the potential that you'll one day want to commit suicide. And of course, it's not limited to television, radio. Last Christmas, there's even a movie about concussion and about the physician, Dr. Bennett Omalu, who was the first CTE scientist modern CTE scientist to present a case, which was a case of an NFL player in 2005. So this is very, very present and available for us, not just as scientists, but just as anybody who has an ear or an eye on media. So what is this? What is chronic traumatic encephalopathy? This isn't new, first of all. Chronic traumatic encephalopathy has been talked about in some way, shape, or form since about 1920. Harrison Martland, a physician, started to look at boxers and noticed changes in their behavior, and in particular some changes in motor behavior in boxers who'd sustained a significant amount of neurotrauma. So this is in 1920, and it was called dementia pugilistica to begin with, and had various names. And around 1940, chronic traumatic encephalopathy was the term that was preferred by many. And again, in 2005, things changed. We had a new case with an NFL player, and what changed was the label stayed the same, but the symptoms and the pathophysiology was quite different. So the symptoms that we talk about now with modern CTE are quite different than what was described historically in boxers by the early physicians who are looking at this. Rather than a predominance of motor signs, we now see a lot more cognitive and emotional symptoms being present in people who are suspected of having CTE. And then when you look at their brains later, then you see neuropathologic changes that support that diagnosis. So this is what current CTE scientists think is going on. Repetitive neurotrauma triggers progressive degeneration of the brain, which causes a buildup of an abnormal protein, tau. They believe that these changes can begin months, years, or even decades after the initial traumatic event, the initial injury. You typically see the changes in behavior, in symptoms, following retirement, and then up to their death. And then, of course, they look at the brains and see if there's any evidence of CTE. So if you've never seen it before, this is a slide that gives you a bit of a heads up about what CTE looks like. So over on the left, we see a normal brain. This is a non-athlete. So this presumably would have been an individual in a brain database who'd never sustained a concussion and didn't have a history in contact sport. Oops. In the middle, we have two different brains from athletes who had sustained neurotrauma, and it's this brown aggregate of protein here that you see here, and quite easily seen over here, that is this tau protein that they're looking for when they're trying to see the pathophysiologic evidence of CTE. So when you're thinking about CTE, think about two things. Think about the pathophysiology, but also think about the symptoms, because it's the symptoms that get people concerned. Well, you know, an athlete's retired, their behavior starts to change. What's the cause of that change? Well, it must be CTE. They had, 5, 10 concussions during their playing career. So from a symptoms perspective, what does CTE look like? Here's an example. An ex-NFL running back. After retirement, sometimes I think I'm going crazy. My mood has changed. So some significant issues with emotional ability. Jenkins began to suspect CTE several years ago when an uncharacteristic dark mood and deep anger began to bubble up at unexpected times. So those kind of statements are fairly typical in, if you actually look at the case data, they're very detailed and interesting to read, and they have a lot of information in it like that. Here's another one, an ex-NFL player. So becomes socially isolated after retirement, was depressed, and attempted suicide multiple times via different means and mechanisms. Had paranoid ideations. Some of it had to do with financial ruin, and eventually committed suicide 11 years after retirement. So remember I mentioned about historic CTE and the primary symptoms being motor in nature. So what a boxer would have looked like, they would have been boxing, and they boxed often. Back in those days, they would have been club boxers, boxing weekly, bi-weekly sometimes, so experiencing a lot of trauma in a short period of time. So one day they would realize they weren't moving the way that they normally would. They've got a foot that's dragging. Their physical body is starting to break down, and then they also start to note these other cognitive signs in addition to slurring and other things. Here we've got a list of symptoms that look very different from historic CTE. As you can see in the list, headache, depression, suicidality is actually one of the stated symptoms of CTE, impulsivity, explosivity, and aggression. So you go down the list, it looks very different from historic CTE. And so that's the kind of thing that modern-day CTE researchers are alarmed about. These people are changing, and they want to know why. So it's led to statements such as these, some fairly bold statements by CTE researchers. So CTE has been described as a neuropathologically distinct progressive tauopathy with a clear environmental etiology. So what does that mean? It means that neurotrauma in 100% of the cases is causing those symptoms and that pathophysiology. So followed up by another one. It's the only known neurodegenerative dementia with a specific identifiable cause, in this case, head trauma. So you would hope, based on those bold statements, that there's some very, very robust science behind this. So to date, there are how many published prospective studies relating to CTE that could generate data to support these statements? How many? Guess. 20? 10? None. There's none. So this is a problem. This is a problem as scientists trying to come up with accurate information for the media, accurate information for parents who become worried that their kids are playing football, they're seeing their heroes falling and being diagnosed with this thing that leads to suicide. They want answers too. So if you actually look at the entire population of CTE cases, you start to be able to disentangle some of this stuff. So historically, if you look at CTE and look at historic CTE, modern CTE, there's 158 cases total in all the published literature. 85 of those can be considered modern. So from 2005 with Bennett Amalu's first case published, 85. If you break those down, this is what it looks like. 20% of those individuals had, at autopsy, pure CTE. Over half of them had CTE plus something else. So if you're making statements about, these people are behaving differently and it's 100% to do with the fact that they have CTE, well, it's not. They have Alzheimer's disease. They have other diseases that we know also affect behavior and cognition and so on. Furthermore, 5%, so 1 in 20, had some form of neuropathology but no CTE at all. And about a quarter of them had all of the symptoms you'd expect to see, and then you'd expect to look into their brains and see this CTE, and they didn't have anything wrong with them. So this one-to-one connection between symptoms and pathophysiology is not as clear-cut and not as simple as it's being portrayed. So why is this the case? How did it get to this state where it's in the media every day and it sounds like there's these serious problems, and if you actually start to look at the data, it's not quite so clear-cut. It has to do with how the cases are selected for the brain data banks and databases that they're put into. So how are they selected? Are they selected randomly? We've identified these aren't prospective studies, so how do you get a brain in a brain data bank that's going to eventually make its way into a CTE study? Well, they're donated, and they're donated by family members that want answers, typically. They see a change in a loved one who had a career in sport, and they start to behave differently, and they want to know why. And sometimes they commit suicide, and that's horrible, and they want answers. So they donate the brain to science, and that's how you end up with a very highly selected number of cases that eventually become CTE science. As scientists, we know there's many problems with looking at that as our primary mechanism to establish what CTE is, how prevalent it is, and so on. So many scientists now are starting to understand this and comment on it and be very critical of it, to be honest. So this is what CT case research, and in fact all of CT science, looks like. So if you look at the circles, independent of whether they're white or red, they could represent all retired athletes in a specific sport. You look at the red ones alone, and they represent the ones that are having problems. They have some change in behavior, they've got some problem with their memory, they've got some issue, and they think that they potentially have CTE. We're only including the red dots in our science. So as scientists, you know that that's a problem. So how can we fix that problem? If we want to do better science with CTE, we should be thinking of it in this way, prospectively. So athletes retire. We should look at a random sample, or even all athletes if the league's not large, and look at them not just in terms of how many concussive events they've had, because then we've already made up our mind that that's what the problem is. We don't know that that's what the problem is because we haven't looked at any other problems yet. What are some other potential problems that can create that symptom set? And these have been studied very, very well. How well you're adjusting following a career in sport in terms of your athletic identity and transitioning from high-profile athlete to civilian. How much chronic pain do you have? How much pain medication do you use? Did you use steroids? These are the kind of things that we know have an effect on physiology an effect on cognition and emotional ability. So ask those questions, document them. The person eventually dies, and then look for the evidence of CTE. Now you've got a record of symptoms, you've got a record of CTE, and now you can look at whether they match up or not, rather than assuming that this pathophysiology that you see in this brain is linked specifically to those symptoms that you saw before the person died, most of which were provided by family members retrospectively. So I know that's very critical, but I'm not alone in my criticism in this area. In fact, there's as many papers out now in the last 2 years being, that type of critical as there are publications on CT specifically. So here's my area of science. Now we're talking about me as a scientist, so let's see if I can stand up to my own rigor in terms of prospective studies and whatnot. Okay, so when we talk about electrophysiology, what is it that we're talking about? We're looking at new ways to assess and manage concussion using electrical activity of the brain. So what I'd like to review today, what's the current state of the science, and are we able to take any of these EEG, event-related potentials, evoke biomarkers, and translate them into something that's useful and practical clinically? Because we've been doing this for quite a while now. We did our first EEG studies and published it in 2000, so it's not like this is new. With some hope, we're moving forward into something clinically useful. So like I said at the intro, we'll look at EEG, we'll look at evoked potentials, we'll look at event-related potentials. So any electrical response that we're recording from the body, some of you know this very, very well, we're recording extracellular potentials. And in the brain, we're looking at axons, and typically axons that are traveling together, maybe in tracts, but maybe between, within a hemisphere or whatnot. So we're looking at electrical change primarily in axons of pyramidal cells, and these extracellular potentials are what we're recording. So if you haven't seen EEG before, EEG looks like this, but you can actually break it down into waves of different frequencies and different amplitudes. Basically, if it's high frequency and low amplitude, those are the types of waveforms that are associated with cognition and thinking. Down here you have high amplitude, low frequency waveforms like delta activity and theta activity that you typically see when you're sleeping in adults. So you take all of these and throw them together and you get this. So you get an amalgam, and we have mathematical procedures for separating the waves into different frequencies and bandwidths. So here we have a number of studies that look at the EEG and the acute phase following concussion, and you can see that there's some patterns that start to emerge. So this is fairly well researched, and some of the studies were done quite a while ago. So you look at theta activity, and you can see that all studies reviewed here, even some that recent ones that include exercise, you see this elevation in this sleeping wave, this wave that you'd expect to see more so while you're asleep in waking adult EEG after a concussion. Not so much over here in the waveforms we'd expect to support cognition. So there seems to be some reasonable pattern here that one day you hope we could parlay that into something useful clinically and come up with some measurements and techniques to do that. So there's a body of data here that seems usable. There's also what I'll call some advanced EEG paradigms, some that take EEG recordings and couple them with balance or some other stimulation. And they're a little bit mixed. Some of them do show some changes in beta activity, but for the most part, I think if you look at the entire, if you aggregate all the EEG studies, it seems like the emergence of these slow frequencies seem to be a predominant result in these studies. So we've done a lot of work, and it seems like there's some consistent pattern here. So how does that compare to some of the other studies with evoked potentials, event-related potentials? So evoked potentials are a little bit different in that you're providing a stimulus for an individual, a very simple stimulus, something that activates a primary sensory pathway or a motor pathway, and you're looking at the timing of the response. So this is an example of a pattern visual evoked potential. So very simple stimulus. As I mentioned, white squares turn black, black squares turn white. You do that about 100 times and average them all together, and you get a response like you see on the right-hand side. And with these peaks, we're looking for changes in amplitude, so the size of the signal, and the timing of the signal. So changes in amplitude, changes in latency. So there hasn't been a lot done with the PVP or P100 response, but there was one study that looked over time. So I guess you could actually consider this prospective, but it's a case study, where over a year, this individual, this young girl, had a concussion, significant change in the PVEP response, and then over the period of months and a year, there was eventual recovery of that response. But there's been quite a bit in the MTBI literature, but not so much in the sport concussion or concussion literature. Motor evoked potentials have been studied a lot lately. So motor evoked potentials can be stimulated in multiple different ways. You can use a magnet, activate motor cortex, and look at some motor evoked potential at some distal limb, hand or foot. You can do changes in posture, changes in balance, and elicit these responses. So these are very popular. This is what it looks like, more or less. You get a coil, you send a magnetic pulse through the coil, it activates primary motor cortex, and then again you record your response somewhere distally. So you can record a lot of different things here. You can look at how information moves through white matter. You can look at the excitability of the motor cortex, and that's what a lot of people have done. You see these downward arrows. This is indicating a decrease in the excitability of primary motor cortex when stimulating before versus after a concussion. So it seems to be a reasonable way to assess the acute phase of concussion as well. There's newer studies that you can see here that are looking at inhibition, actually how concussion can affect inhibition in motor pathways as well. So again, some fairly robust, fairly decent findings here that seem to be following some sort of pattern. Event-related potentials are cognitive in nature, so they move beyond a simple stimulus, like a simple pulse, electromagnetic pulse, or a simple flash, to something where the individual's doing something. So they're performing some simple cognitive task, and you're recording from the brain while they're doing that. So one of the most prominent is called the N2P3 response, or P300. It's been used since the 60s for a variety of different things. The various peaks are interesting, but the one that's most interesting is the P3 response. So the amplitude of the P3 response is thought to be related to attentional resourcing, and the timing or latency of the response has to do with how salient the stimulus is, how you're categorizing the information, and essentially how you're identifying it as important versus not important. So this is a response that is generated from multiple cortical and subcortical areas, so it's not specifically located in one area of the brain. The paradigm that you use to elicit it is called an oddball paradigm, which is essentially there's an infrequent stimulus and that's embedded within a series of more frequent stimuli. So it could be something like a high tone embedded within a series of low tones, and it's randomly placed in those low tones. So you're essentially thinking and listening for this odd stimulus and counting it or responding to when you see it. So when you do that, you generate an additional peak, which is this P3 response that you see over here. So when you're just listening to the more frequent tones or visual stimuli, you don't get that peak. And when you pay attention to this infrequent odd stimulus, you see it there. So there's been quite a bit of work done with this in the auditory modality and the visual modality. And really what you're seeing with these studies is some fairly consistent findings in the acute phase following concussion, with a reduction in P3 amplitude and some studies showing an increase in latency. But the primary finding here is a temporary reduction in P3 amplitude. These are data that we've collected this has been presented but not published. So what you're seeing here is P3 responses at baseline, so preseason baseline being large, following concussion, immediate 2 weeks and 1 month. You see a dramatic reduction in amplitude with some recovery up to 1 month, but you can see there's quite a gap between the baseline and the recovery at 1 month. So that's not been published in a peer-reviewed journal yet, but hope to soon. Visual N2P3 latency, similar kind of things seen here in the acute phase. So a reduction in P3 amplitude primarily as a consistent finding. So with the N2P3 response, you're seeing something that looks fairly robust, fairly consistent, and fairly well researched. If you're looking for cumulative effects, so not in the acute phase, you're looking at people who've sustained a number of injuries, and you're not looking immediately following the injury, but maybe 9 months or a year following. It's not the amplitude that seems to be the most significant indicator, it's the latency. So for those individuals who've had a number of concussions, you want to see the change in their brain that's most substantial. The one that lasts, essentially, seems to be an increase in the P3 latency. So these are data that we did publish showing a fairly substantial increase, about a 40 millisecond increase in latency in BCHL hockey players. And this shows that with 3 or more concussions, you get this substantial increase in how long it takes your brain to process information, even in very young athletes. And not only that, those increases in latency correlate to changes in memory, changes in concentration, changes in their ability to think. So they have some basis in the symptom profile as well. More recently, there's been a new technique called brain network activation that combines EEG, event-related potentials, and it uses machine learning algorithms to come up with a unique network of activity that's related to specific tasks. And the hope with that was that you'd be able to create a database that represented how we process information and then look at deviations from that complex database. It looked good early on. There was a study that was published by Contos and coworkers that looked at people who have chronic migraine problems not related to concussion, and it was able to separate migraineurs from non-migraineurs. There's a case here that was a concussion case that they used to illustrate this technique, and it looked like it had some promise. But as you can see down here, this year, there's been 2 studies published that basically indicate that it's got too much, that we have too much variability in how we process information to come up with a common pattern that you can then look for deviations from. And that's what the second study here showed, was it doesn't discriminate between concussed and non-concussed individuals. So we just reviewed a lot of EEG information, electrophysiologic information, about 45 studies. How many of these studies were prospective? Guess. None. In other words, we didn't measure them at baseline. I mean, the one that I showed you was, I guess, as close as you could get was the PVEP study. They didn't have pre-injury information on that girl. They just measured her over a year, so you could argue that that one is. But in terms of how other studies have been done, like the ones Patrick showed, baseline, injury, follow-up 1, follow-up 2, follow-up 3, not been done. So this needs to be done. We need to spend the time, the money, whatever it takes to collect a lot of information at baseline and wait around until people have concussions and then follow them up. We need to follow them up in the immediate phase, 1 week, 2 weeks, and so on, to see how the brain changes with concussion and see how long it takes for the injury to, for recovery to occur. We need to do this because if we don't, there's no way we're going to be able to easily move from individual or from group differences, which is what I showed you, to looking at how you can manage an individual, because that's the goal. That's why we do this science, is to one day be able to say, hey, you've had a concussion, come to my clinic, we're going to run you through some tests and definitively tell you whether you had a concussion or not, and how severe it was, and how long it's going to take you to recover and get back to play. We haven't done that yet. We need to do that. And if not, we're just going to be continually doing these group studies, looking between groups and coming up with the next best new novel technique that never gets us any closer to something that's useful clinically. So why do we need more prospective electrophysiologic studies on concussion? Because there aren't any. If we want to use these techniques and procedures, we need to do this. We need to spend the time, spend the money. Prospective studies are a necessary first step. So that's all for me. Thank you. The CT information will be part of what I'm publishing hopefully soon. It's more robust, a way to look at CTE that I'll hopefully publish in Medical Hypotheses. So thank you very much.
I actually interned at the Royal Jubilee Hospital here in Victoria in 1981, and Michael Saunders' father interned with me, and we're old friends, so I have some old connections to Victoria. I'm happy to be invited to speak here, and I think there was a good segue provided to me by my two predecessors in saying that the world of research labs and the clinical world is kind of different in terms of how we have to look at the data and apply it. I don't market any of the devices that anybody is using, and so I just get a salary from Children's Hospital, and I'm appointed at the UBC in the Department of Pediatrics. So I want to talk a little bit about some of the things that, based on recent reviews of lots of evidence that have been published, end up in the hands of clinicians, including myself, who on the one hand are engaged in collaborative research on concussion related to sport, and on the other hand end up having kids referred to us by the time they've been through the first few weeks of not getting better, and somebody wonders whether a neurologist should see them. So some of what I have to do has to sort of take information one way and some in another way. You may or may not be aware that there was recently published a very extensive review that's taken place over the last 4 years of pediatric mild traumatic brain injury data. And the thing is that if a kid slips and falls on a playground, it may be counted in the statistics as a mild TBI separate from sport-related concussion, which may be counted in another database. But to the healthcare system, these kids look the same and deserve to have similar standards applied. So I'm listing here, or showing here, the front page of this big document, which was co-authored by Keith Gaetz, who's at the University of Calgary now. I guess the big point that has to be made first is that mild traumatic brain injury and concussion is still a clinical diagnosis. And so the way that we as pediatricians and neurologists and sport medicine physicians are trained to do diagnosis is, first we take a history. So we really do want to know what was the event history, and is it compatible with there being some kind of mechanical force applied to the brain. And then we want to know, in relation to that, if there's evidence based on the person's symptoms and the signs that we can see by doing an examination, if there's evidence that function of the brain was disturbed after that force is applied. Then the third thing we have to do is to ask the question, well, is there another explanation for it? For example, if somebody is slurring their speech because they're drunk, intoxicated with a drug, we may want to consider whether to attribute the slurred speech to the concussion or to the trauma. So this is where our terminology differential diagnosis comes in. Have we thought about the combination of explanations that may go into it? It's quite often the case, though, that somebody will arrive on the doorstep of a physician with the feeling that they really need to know if this is a concussion, so can't you just order the scan? And I didn't put electrophysiological tests up there, but I could easily add them in. And This is a hard part for physicians, is that if we look at the evidence base that we have and the guidelines that we work with, they generally conclude that we shouldn't be doing scans. So we have to kind of be confident that we're doing it the right way. And the way that that's been done over the years is by setting up prospective studies that have involved emergency departments and pediatricians at children's hospitals in various different places who gradually come up and formulate decision-making rules, such as one that is applied at most children's hospitals and, for that matter, most emergency departments for determination of whether a person ought to have a CT scan done after a head injury or not. There are a number of circumstances that, taken together, would allow a person to conclude that a CT scan is likely to turn up something that is important enough to require neurosurgical intervention. But really, children with mild traumatic brain injury have CT abnormalities found only in about 1.9% of the time. And so we have to be careful that we aren't subjecting too many people to unnecessary CT scans, and that's why these decision rules have been applied. It is the case that if somebody has depressed level of consciousness after a TBI, they're more likely to have a justified reason for getting a CT scan done. It is the case that if a systematic approach is followed and published decision-making rules are followed, that the confidence in being able to conclude that somebody doesn't need a CT because they're not likely to have an important intracranial abnormality are applied. So really the recommendation that is out there that we're all expected to follow is that we not obtain routine CT scans unless there's a high risk of a clinically important intracranial injury. And risk factors that come into the decision-making rules include infants under 2, vomiting, loss of consciousness, a variety of mechanisms of injury, headaches that are getting worse, amnesia, and the other things that you can see listed here. So that's one point. And so there was a very large study in the United States that Nate Cooperman, a friend of mine from my days in Boston, was the lead author on. They studied 32,000 kids at various U.S. emergency departments to come up with this decision-making rule to decide whether kids with closed head injury should have a CT scan or not. And Glasgow Coma Scale 14 or other abnormal mental status or signs of basilar skull fracture, CT yes, loss of consciousness, vomiting, severe mechanism of injury, severe headache, yes, but otherwise, no. And so they published the sensitivity and specificity and so on related to this, and it's quite high. Meanwhile, in Canada, another study involving nearly 4,000 children was done to derive another rule in parallel that's called the CATCH rule from the Pediatric Emergency Research Consortium. And you can see here that it has a number of overlapping parameters, but some that are a little bit different. So really, the next slide here indicates that currently healthcare providers are advised that if there's a reason, we should discuss the CT scan with the person. But beyond CT scan, there's really not sufficient evidence to recommend MRI scans of any kind, including the advanced ones, in the routine care of children with mild traumatic brain injury, including concussion. And the same applies to SPECT and to skull X-rays. So that's the first thing, is when somebody comes to me and says, I haven't had a scan, then I go through the checklist and I try to discuss with them whether they really have a valid reason to expect that they would have an actionable intracranial injury, and if not, then I try to talk them out of it. But I also suggest to them that there's a lot of good research going on, and that if they want to get a scan, signing up for a scan that involves imaging is a good way to add to the scientific literature, but not necessarily to the decision-making at hand about their care. So then the guidelines go on from this document to tell us that there's some things that we should do for counseling, and there are things that we should do for giving people advice about the contribution of premorbid conditions. And I think I'll spend a minute or two about this, because I think it's really important that we actually do largely reassure people that things are going to get better over time. And I think that when somebody comes in and they've had a nerve injury, a facial palsy or something from a nerve injury, I will quite commonly tell people, look, this is likely going to get better, but it may take time. You may see a lot of improvement over 6 weeks, but in some cases it might take a year or two before you get to the full extent of the recovery you're going to have with a facial nerve injury. Well, I'm now starting to say the same thing to kids who've got traumatic brain injury. Early on, we should tell them that there's quite a lot of variation from person to person, and even though the luckiest ones have a fairly quick recovery from symptoms that trouble them, don't feel terribly despondent if your recovery trajectory takes longer, because we can't predict it in the beginning as well as we'd like to. And there are a number of premorbid conditions that we know are associated with a higher probability of having a delayed recovery, one of which is having a previous episode of mild traumatic brain injury. But in general, if you have some neurological condition, or if there's something not quite typical about your brain and the way your brain works, you're a little bit of a setup for a delayed recovery. And in addition, there are family and social factors that seem to contribute as well to that. So really we should, I think, be giving this message to people as well, if we know up front that somebody has had a traumatic brain injury and they've also got ADHD or learning disorders or history of migraine headaches or family history of or family difficulties with family distress, we may counsel them that it might take them a little bit longer than average. Same thing if we know that somebody's had a seizure disorder or something else with abnormalities on a pre-existing MRI scan. Well, this is an abnormal brain from the get-go, relatively speaking, so it's higher risk for delayed recovery. So I've already mentioned the thing about prior MTBI being a risk factor, and I was so distressed to hear that Sidney Crosby is out again after being really distressed when I remember seeing what happened in the two games that were played close together when he was at the beginning of the Odyssey. So interesting that on the NHL website, when they announced it, they said that he was injured during practice, and then the next piece up there, which is why I put the side in here, is it said no timeline for return. And so I also use this when I'm talking to parents and kids. If a professional athlete like Sidney Crosby, with the resources of the NHL or behind him, can't buy a scan and a cure to make them better in a week or 2 weeks or whatever, and they have to announce to the public that there's no upfront timeline that can be given for return, we have to wait and see and make sure that he takes care of himself and that we help him take care of his brain. That's really all that can be said for Sidney Crosby, so it's all that we can say for our kids. So in the aftermath, we need to monitor them and anticipate that most of them in a number of weeks, 2 or 3 weeks, if not less, will be a whole lot better. But occasionally they take longer, and if they take longer, still by and large within a year or two they end up getting better. but they need more help because it's going to have more impact on their life. So post-concussion syndrome is I think, the thing that becomes a worry when symptoms persist beyond 1 or 2 or 3 months. And so the emergency departments across, the pediatric emergency departments across Canada have also looked at that in a collaborative way with 3,000 patients, following them up serially, looking for post-concussion syndrome as an outcome, and came up with this 5P study predicting persistent post-concussion problems in pediatrics. That's the 5 Ps. But the thing about it is that by looking at risk factors for post-concussion syndrome and kind of quantifying things with a prospective study with large numbers, we know that adolescents, teenagers, are a little bit higher risk than the younger kids. Female gender is a risk factor. Prior concussion, migraine, difficulty with balance, headaches, noise sensitivity, fatigue. These are the factors that they actually came up with from factor analysis, put into their model, and used for their predictions. And here's how it worked out. If you get categorized in the high-risk category, your probability of post-concussion syndrome is I think that's 4 times higher than if it's low risk. So these rules are actually quite helpful, and this is how they're being validated. I think it's important that we do provide education and reassurance. The second point on this slide here is that we should counsel them to rest in the beginning, just as we'd advise somebody to rest for a little bit after they've had any other injury. But then we want to get them going after that, and along the way make sure that they get support that they need and get back to their job description, which is largely going to school. So there are a variety of things that physicians and healthcare providers are encouraged to do, which is to, if someone's been encountered in the emergency department and they're being sent home, they get a head injury sheet so that if they have a depressed consciousness later, they're brought back. These sorts of things, bad headaches, are brought back. Prevention of further injury is important. We know that kids who've had concussion are not only more likely to have further concussion if they return to activity soon, but also injuries of the rest of their body. But one thing that I think is not appreciated by athletes is that if I tell somebody you shouldn't go back to playing full contact hockey, ice hockey, yet till you're cleared, okay, so they'll do the graded stepwise thing that we all talk about. But then if they're adolescent males and they're playing with they're doing something with their friends, they'll get bashed into the locker or something like that, and they'll bang their head, or they'll go out and run bases, a sudden burst of maximum sprinting because it's not within the boundaries of exactly what they were told was okay or not okay. So I also tell people that in addition to not going back to things that we know will put them at increased risk, the full contact gameplay, I specifically suggest that they be very careful about horseplay. And I don't know if others of you have experienced that, but I've had a number who've come back with re-injury, and when I take the history and say, well, what exactly were you doing when you're making good progress until something? What exactly happened then? And it's the out of bounds horseplay sometime that comes up. Whoops, sorry. So rest. Yeah, we don't want kids put in a dark room and the key sort of thrown away and come back and check on them in 3 months' time. For a few days, if they have a lot of photosensitivity and headache, that's probably where they're going to want to be anyway. Most of the kids that I see who have bad migraine headaches with photosensitivity they put themselves in the quiet, dark room because that's where they feel best. And I think that sometimes is the case for kids after they have a traumatic brain injury. Is they know that that's what the only thing that they can do for a few days. But after that, we need to gradually get them back on their feet. And I think one thing that came out of this report that I'm happy to see is there isn't evidence to tell us, good or bad, that strict rest is a benefit. But at least that means that it isn't justifiable for anybody to say that you must rest and not do anything until you're entirely symptom-free. This makes me want to go back, though, to a gap that we have in data. We don't really have as much prospective data as we'd like to have about what happens with mild TBI. And certainly from years ago, the studies that were done at UCLA on severe traumatic brain injury were helpful in telling us about this cascade of events that happens after a concussion, but particularly about the things that happen in the first few days. And I think that Patrick was alluding to this, that in severe brain injury, there for the first few days is, or in the first 12 hours or so, there's hypoperfusion, and then there's hyperemia. And so in the intensive care unit setting, we're used to seeing brain swelling. And if a kid comes in with a stroke or a traumatic brain injury and they're doing okay, I always warn parents that things might get worse until they peak around day 3, and then if whatever they're at day 3, they typically dial back somewhat after that. So there is this hyperemia phase that we see a lot in the hospital, and then after that, there's a prolonged recovery phase, and I think that's most attributable to problems in the microcirculation that take longer to recover. And the time frame for that is really not known in mild TBI. The other thing is that we don't know about the first 12 hours in mild TBI because nobody does these fancy studies measuring cerebral blood flow as they did in these folks with transcranial Doppler and cerebral blood flow measurements. Nobody does this in the first 24 hours of mild TBI. So if we do the stepwise protocol, then we want them to get back to school, and we probably have to get some communication going with the school. And then I'm going to come back to this slide, which is from the American Academy of Neurology's guidelines from 2013. The best advice that we could think of giving to athletes who are collegiate athletes or older adults, we should dial back a notch and be more conservative with kids, because the adolescents are a little bit more at risk for things like second impact syndrome, rare though it fortunately is, and so I think that if there's anything that I really like to emphasize to folks, it's we'd like to get your son or daughter back to being active and fit, but not necessarily at risk for or exposed to risk to be re-injured until we're very comfortable that they are fit and agile again and thinking clearly and doing well at school and doing all their specific skills really well. Occasionally kids need help from a psychologist for cognitive difficulties that they have afterwards, and I'm going to also say that sleep difficulties are quite common, and I think there's evidence that sleep is restorative and does perhaps help to clear some of those tau proteins that arise, and so managing prolonged headaches and managing fogginess and thinking about whether sleep is a contributor to that, I think is something that we should do more often. So that's my obligatory recitation of the rule book that I have to sort of try to follow. There is interest, obviously, in doing advanced neuroimaging, and some of the areas of the brain where fractional anisotropy changes of diffusion-weighted imaging have most often been found are in the corpus callosum, and so this is a picture from one of such studies in children. There are many studies of this type. They're certainly not in the realm of readiness for clinical application, though, because we have a lot of kids who have incidental findings, and we have a lot of transient findings. If we took somebody off the street who didn't have a concussion, we might find that they would have some of these changes. So we're, I think, still in the information gathering stage. I think, Patrick, you mentioned this study, Logan study, but thinking about the three-phase severe traumatic brain injury blood flow, hypo to hyperemia, the finding from this study was reduction of CBF in that sort of chronic phase. And there was another study that was published by Karen Barlow and the group in Calgary that instead of demonstrating a reduction, seemed to suggest that there was an increased cerebral blood flow from their study. So those are, I would say, contradictory findings. And you also mentioned this one, Patrick, so I'm not going to bore everybody except to come to the results of this one. You showed the pretty pictures of the insular cortex, which is quite important in autonomic control, and that this took some time to recover, but was decreased in the aftermath of concussion. I thought that was kind of interesting as a prospective study. There's that depiction again. Just to emphasize that this was verbatim from their conclusion, they indicated that theirs was the first prospective study with mild TBI, because as Mike was saying, there had been 0 prospective studies on mild TBI that were done the way with similar imaging techniques done on severe TBI. So last thing I'm just going to mention is that the pediatric or the Children's Hospital Emergency Department Research Consortium has decided to go beyond the 5P study now and band together to prospectively study kids with mild traumatic brain injury and do serial imaging studies as well as behavioral and cognitive studies. So the Children's Hospital in Vancouver has signed on to that, and my colleague, Dr. Quinn Doan from the emergency department, is going to run the emergency department aspect of it, and I will run the imaging part of it. We're going to enroll 8 to 16-year-olds with either mild traumatic brain injury or orthopedic injury, and it'll be a prospective concurrent cohort study. And the total sample size is going to be 1,000 kids across Canada from BC Children's Hospital, the Alberta Children's Hospital in Calgary, Stollery Children's Hospital in Edmonton, Children's Hospital of Eastern Ontario, and Saint-Justine in Montreal. Keith Yates, who let me borrow this slide, was kind enough to identify each of the cities by a corresponding team logo. The study has the acronym ACAP, and we've we are just starting in the next month to enroll subjects at Children's Hospital, and they're just starting in Calgary. So I would say stay tuned. I'm hoping that this will help fill in some of the gaps. So I'm sorry, but I haven't looked at my watch because I was terrified that I was going to go over time, but I think I'm at the end. So thank you.
I think we're going to have some time to get some questions. I just want to summarize a little bit, because what I think you've heard is that we know automated mechanisms in the brain are being affected. It's just hard to measure. But once we get those measured, it'll be valuable. What Bruce had presented about that neurometabolic cascade is something I use with athletes to try to explain to them what the injury is so that they kind of get a picture, because you can't show them a radiograph that's going to show them that. And Bruce has also talked about that. There is no imaging. What no one talked about, and what I want you to think about, particularly as young researchers, is there's a commonality between asbestosis, mesothelioma, smoking and lung cancer, and concussions and CTE. And that is that there's been a huge, huge organization going forward trying to minimize liability by denying those things, those connections. So all the science that predates some of this current research in concussions was manipulated by physicians and scientists that associated with the NFL and other things. So if you want to read about that, you can read about the League of Denial, which is the background of the Will Smith movie. In the area of asbestosis, the asbestos companies trying to minimize liability, and as you're aware, tobacco companies did the same. So there is a commonality in this very devastating injury in some instances. And so suppression of data has been a bit of a problem, and Michael showed you that there's a gap in research, and that's a big problem. Return to play and return to school is a major issue for us clinicians. There is a lot of discrimination to people with concussions because, again, there's no casts, there's no sutures, there's no Band-Aids. To the point where in Ontario, the school districts were imposed upon by the government by a bill making them liable to manage concussions and not discriminate against students missing school because of their recovery time. So when you have to implement a bill in provincial government to tell teachers and educators to back off and let kids recover, that tells you a lot about what's going on in this particular disorder. So I'd like to open it up. If you've got any questions for any of the panel members, that'd be great. I'm sure they're all willing to answer anything you've got. So the question was, what any comments about concussion apps?
Well, I'm a coach, and I've coached a long time, and I know coaches are sort of creatures of habit. I think maybe for younger coaches, they might find something like that useful. There's still a lot of coaches who are in complete denial that there's any kind of problem at all, and then you get sort of middle of the ground coaches that are thoughtful but are following the guidelines, sort of the standard guidelines. So I think it really depends on the individual. I think it just depends on whether they're tech savvy, whether they, the younger generation is, moderate stage coaches probably aren't. So there may be some promise to that. The work that I've seen is sort of mixed on it, though. So
there was another hand up. Or do you mind using the mic or shouting? Because otherwise I got to repeat what you're going to say. Okay.
there are two current large studies. One is called the UNITE study, and I can't remember the acronym for the other. They're being performed by the same researchers who've done most of the work with some additions. The problem with both studies, though, is that they're not prospective, and they're not randomly sampling all retired athletes. If you're only looking at one type of athlete, you're only going to come up with one type of athlete to study. So are there any properly designed, in my opinion, studies that are on the horizon or ongoing? No, there aren't.
I have to take a shot at that. One of the things that's challenging is that the autopsy rate has gone down over the years, and people who don't perceive that they have a serious problem, their family members aren't likely to want to consent to an autopsy. So while for academic discussion purposes we can sit up here and we can say what we need to do is to make sure that every single NFL player is in a registry and at death undergoes an autopsy, it's not likely going to be the case. So the people who have the problem are going to be the people who have the motivation. But on the reverse side, if you take a population of people who are NFL football players and find that they have a pathology that doesn't proportionally exist in the rest of the population, then I think there's something that you have to pursue. So I'm aware of one other thing that might be relevant here, but not specific to CT, and that's that there is a kind of a PET scanning tau-labeled radioligand that is perhaps coming onto the scene in the next couple of years. It's being advertised at least, so that may help. That is, if people won't undergo an autopsy, perhaps they'll go an in vivo test if it can be demonstrated that it shows the pathology without having an autopsy.
The sad part about that is that some individuals committing suicide, when they know they have something wrong, have gone to the extent of shooting themselves in the chest, not the head, to make sure that their brain is available. Others have just donated their brains ahead of their deaths and their suicides to make sure that they're available, but otherwise there's nothing for us.
Is there any evidence yet in determining, so we talk about in emergency management, the golden hour, is there anything like that in concussion-related recovery where someone who's trained in sideline assessment pulled the athlete or child, or whichever situation is, out of play immediately versus maybe delaying that? Is there a difference in the timeline for recovery? Have you evidence on that?
Actually, we don't have a lot of evidence on that. Intuitively, we would think that holding them out for that longer period of time, certainly as Bruce mentioned earlier, there's this problem of a syndrome that can actually be created, secondary impact syndrome. Some don't like that term, but I believe that it really does exist. It doesn't happen very often. And so the longer that we can actually take the individual out of play, certainly the recovery is going to start to occur much sooner and much quicker without the added stress potentially of getting into another injury.
Some professional programs have mandated time frames which athletes have to be pulled. Some programs, World Rugby, for example, has a system of spotting mechanisms associated with concussion without diagnosis that you can pull athletes without team influence. So there's a variety of things that are available, but there is no mechanism to determine how long.
Just one added comment, for the athlete who goes down and loses consciousness immediately, it's simple. That person qualifies and has to come off the field, and they're done. For the person who sustains a blow and is out of it, neurologically compromised, that's pretty clear. But there is data that show that a substantial number of people don't have their manifestations immediately. It takes a bit of time for them to manifest. So the golden hour question is it's applicable to some, but for others, if you saw like what you think is an at-risk hit, it's possible that if you did the sideline assessment, did everything, the person would pass, but hours later or the next day would meet criteria if you reapplied them. So I think we have to kind of bear that in mind as well.
So if you do immediate assessment at the sideline, and someone could have been knocked out even, but often you won't find anything, whether you use a SCAT form or any other assessment, so it gets back to clinical judgment. So organized sports at higher levels will have that ability to pull an athlete, regardless of the findings of that on that assessment, that head injury, mandated head injury assessment, for a repeat assessment in 24 hours. And they're watched between that period.
There are various devices being tried out now to try to mitigate the force that will cause a concussion. Are any of them showing any good evidence or working?
Well, first, I'm going to answer the first question. There is no helmet in this world that can prevent a concussion or minimize it. Your brain is floating in your skull. Unless you're going to put foam in there somehow in between the skull and the brain, it's not going to happen. Dr. Neary is working on evaluating this collar, let's say, that's supposed to increase cerebral pressure and thus minimize concussions. Do you want to answer that?
Yeah, as Steve just mentioned, we can't prevent a concussion by putting a helmet on. The helmets today are certainly significant better than what we had years and years ago, and we are preventing lacerations and fractures to the skull, but we're not preventing concussions. And as Steve has just alluded to, there's a device out today called a concussion collar. Some have criticized the fact that we're trying to put pressure on the jugular bilaterally so that we can increase the volume, even a very small amount of volume within the brain, to increase the size, or I should say to reduce the amount of movement of the brain within the skull, because that's what a concussion is. It's a movement of the brain within the skull. Yeah, and actually, one of my graduate students, Ryan Deck, presented some information today, this morning on a poster, talking about the changes that can actually result looking at the collar. Yeah, and I know Dr. Bjornson has something to add to this too.
Well, I think that the two pieces are complementary pieces of information. That is to say, there is, as you mentioned, value in having a helmet in that it reduces the likelihood of getting a skull fracture. Or if there's going to be a direct impact, you know, a helmet that somebody wears when they're a batter protects them from having an epidural hematoma from a fracture, you know, from a ball coming to the side of the head. So there is value to the helmet, but the momentum of the brain in motion when a person goes down and lands on the ground is of course what produces those contre coup injuries that we see on the opposite side of the brain, and that is from the motion of the brain inside the skull. So I think it has to be said right up front that Patrick's student is working on an entirely investigational, hypothesis-driven kind of project, which is ready for the evaluation phase, but it's not, I think, a good idea for any of us to imagine that we should be recommending it before it's well studied. And I was telling Patrick that I have a pretty hot email trail from some very substantial arguments among some physiologists who are publishing critiques about whether this research should even be permitted, which I think, of course, it must be permitted because it has enough physiological basis. But I think that you all probably know that the literature gets pretty hot in some areas, and this is one of them.
Oh, there's another one. Go ahead. You're at tag.
Well, first, thanks for those really informative talks. I don't are the changes we're seeing, say, in latency, are they mitochondrial, are they synaptic, are they genetic, all of the above? Can we actually measure any of that stuff? Is there a way to know that?
In extensive animal modeling, it's been demonstrated that these are primarily injuries to axons. They're not torn axons, they're stretched initially, and you see a metabolic cascade that occurs over hours and days where axonal transport in the axons causes some swelling on either side of the stretch and eventually a dissociation of either end of the axon that can result in two outcomes, one stability of an injured cell and the other death of the cell. So in humans, that time frame is longer. In rats and mice, it's shorter. So a lot of the initial work, and there's extensive work that's been done on it, it's been shown that this is an issue at the axonal level. If there's some recovery of the axons, what you're left with primarily are a lower number of axons, or axons that have sort of mended themselves back together, and you're left with this compromised system. That, and add that there's been some studies about reduction in dendritic spines with successive concussions. So on one end, you have a reduction in the ability for a cell to communicate with other cells, and then once the cell's been activated, you have a slowing in the transmission of information along an axon that's repaired itself. There's definitely studies that show that that's involved. But in terms of what the primary injury is, as Patrick said, this is movement of the brain within the cranial vault, causing a stretching of white matter. Potentially one cell amidst 100 non-injured cells is what it looks like in animal models. And if you have enough of those, then the 1 in 100 becomes 2 and 5 and beyond that.
And actually, Dr. Bjornson did provide a slide of that, looking at that neurovascular, our neurocascade, the chemical changes that occur as a result of what Michael just talked about, the stretching of those axons. It disrupts calcium, right? And then with calcium, then, of course, we've got a whole cascade of events that can happen.
Any other questions?
I just want to add to that.
Oh,
I think we
sorry.
shouldn't forget the astrocytes and the endothelial cells, because when you're talking about the neurovascular unit, it's all of them. It's the astrocytes that are maintaining the integrity of the neurons and the endothelial cells that are all interacting together. And so, quite typically in the aftermath of stroke and traumatic brain injury, there's swelling of the feet of the parasites, and that may affect the caliber and the vascular reactivity and a variety of other things. And also there may be a switch from oxidative to anaerobic metabolism and flux of excitatory amino acids. And so I think it is a complicated thing that we can't directly study all the parts of simultaneously in humans.
Certainly further to that, there's suggestions. There's a release of nitric oxide as well, which again has its endothelial consequences.
I'm afraid I think we're going to have to end it there because we're running over time almost. Any of you interested in reading more, there'll be an international consensus statement coming out next month, come out every 4 years on sport concussion, and hopefully that'll provide us some more interesting reading. I'd like to thank our speakers for excellent presentations today, and thank you for coming.